In my previous video, I had discussed about the step 3 of protein sequencing, that is identification of N-terminal amino acid. In this video tutorial, I am going to discuss about the step 4 of protein sequencing, that is identification of C-terminal amino acid. Now, to identify the C-terminal amino acid, we have two different methods. One is enzymatic method and second one is the chemical method. So, first I will discuss about enzymatic method. So, enzymatic method. In this method, we use special type of enzyme, that is carboxypeptidase. Carboxypeptidase. Carboxypeptidase is a member of the exopeptidase. That means it acts on the particular peptide chain from the outside. So, suppose this is the polypeptide chain with its N-terminal and this C-terminal. So, as the name suggests that this carboxypeptidase, what it will do? It will remove this carboxy terminal amino acids. Now, once this carboxy terminal amino acid is removed, what will happen? New amino acid will be exposed on this C-terminal side. So, in the second cycle, this second amino acid will get removed. On the third cycle, this third amino acid will get removed. These amino acids are removed by the breaking down of the peptide bond. So, if we draw a graph in which on the y-axis, if we take a time, and on the x-axis, we take a concentration of free amino acid, in reaction mixture, So, what will happen? Suppose this carboxy terminal amino acid is the alanine and then this is a valine, suppose then this is the methionine. Then what will happen? If you mix this carboxypeptidase with this polypeptide chain, so first free amino acid concentration of this alanine will increase. So, first alanine concentration will reach at the peak. So, this is graph for alanine. Now, why alanine is first to reach its peak concentration? Because it is the first amino acid, it is removed first. Then secondly, this valine will get removed. So, slowly valine concentration will also increase and it will reach up to the peak. Okay. So, this is the valine. Here this is the peak of alanine, here this is the peak of valine, right. So, first alanine and then valine is getting its peak. Then as a third peak will be of the methionine. So, this third peak, so here the peak is there for the methionine. So, if we follow the time course of the concentration of free amino acid in reaction mixture, then we can able to identify the C-terminal sequence of amino acid in any peptide chain, right. So, like this, it seems very straightforward and very simple to determine C-terminal amino acid in any peptide chain. But there are two different types of problem. Okay. So, let us discuss about the problem 1. What is the problem 1? The problem is that we thought that carboxypeptidase, they remove all amino acids from the C-terminal side at the same rate, okay, which is not true. Suppose this alanine is removed at very slow rate and the second amino acid valine, it is removed at very fast rate. Then what kind of problem it will create? See, here alanine is removed at very slow rate. So, what will happen? Alanine will gain its peak at somewhat later time. So, this curve will shift to this right side, and if this valine is removed at very fast rate, then what will happen? This curve will be shifting to the left side. So, once this curve shifts to the right side and this curve shifts to the left side, then alanine and valine, both graph will be almost overlapping, and at that time it will be very much difficult for us to determine which amino acid released first. So, this is the first and major problem, right. The second problem is regarding the specificity of particular enzyme. So, second problem, that is problem 2, is with regarding the specificity of enzyme. Now, just like all other enzymes, this carboxypeptidase, they have also specificity. Now, to understand that, suppose this is the polypeptide chain, this is N terminal and this is the C terminal. Suppose this is the C terminal most amino acid, then this is the second one, then this is the third one, and then this is the fourth one. So, we will call this amino acid as a amino acid N. This becomes amino acid N minus 1, this become amino acid N minus 2, and this become amino acid N minus 3. Now, our carboxypeptidase, we know that carboxypeptidase removes first this amino acid. How it is removed? It is removed by breaking down this peptide bond. Now, as you can see, while removing this amino acid N, this peptide bond must be broken down, and in this peptide bond formation, two amino acids are participating. So, amino acid N minus 1 and amino acid N both makes up this peptide bond. Okay. So, this carboxypeptidase, its specificity lies in this peptide bond, and indirectly this peptide bond, as it is dependent on these two amino acid, the specificity of this enzyme, it is dependent on two amino acid, and this amino acid is the N terminal side with respect to this amino acid, which is C terminal side. Okay. So, now from the bovine pancreas, we can isolate two different carboxypeptidase. One is carboxypeptidase A and second one is the carboxypeptidase B. So, with the help of these two enzymes, we will try to understand specificity. So, carboxypeptidase A, it can break down all the peptide bonds except those peptide bonds in which on the C terminal amino acid is arginine, proline or lysine. So, whenever any peptide bond on the C terminal side, if there is arginine, proline or lysine, this carboxypeptidase A, it cannot act, right. Carboxypeptidase A, it cannot act. And this carboxypeptidase B, this also can break down all the peptide bonds except those peptide bonds in which the C terminal side of amino acid is arginine or arginine or lysine. Okay. So, having arginine and lysine on the C side of this peptide bond, this carboxypeptidase B cannot act. What about this amino acid on the N terminal side? So, carboxypeptidase A and B, it can break down all the peptide bonds except those peptide bonds on which N terminal side they have a proline as a amino acid. So, A and B both will not break the peptide bond having a proline on the N terminal. So, by this specificity problem is clear that particular carboxypeptidase cannot break down all the peptide bonds, right. So, but even with these limitations of problem 1 and 2, we can identify several amino acids on the C terminal side. Now, what happens? Certain peptides are resistant to action of this carboxypeptidase, as I just explained you over here, right. So, in that case, we have to use chemical method. So, our second method for the identification of C terminal amino acid is the chemical methods. So, this chemical method, it is known as hydrazinolysis. Now, in this what we do, suppose this is the polypeptide chain, this is N terminal side and this is the C terminal side, and suppose these are the amino acids of this peptide chain. This one is the N terminal amino acid, this is the C terminal amino acid, and rest all rounds are the intermediate amino acid, and this is the polypeptide chain. Now, when you treat this polypeptide chain with the hydrazine at 90 degrees Celsius and for 20 to 100 hours, so it is very long time, and in the presence of one catalyst, that is mild acidic ion exchange resin, resins. Okay, this will act as a catalyst, it will speed up the reaction, and still even after speeding up also we have to wait for 20 to 100 hours, that is between 1 day to 5 days, right. So, once you treat it like this, what will happen? All the peptide bonds will get broken down, and not only broken down, but what will happen? All these amino acid will be labeled with the hydrazine. all the amino acids are labeled with hydrazine except for the C terminal amino acid. So, let us label all the amino acids with the hydrazine. So, this red line represents label of hydrazine except for this C terminal amino acid, right. So, hydrazine binds, or you can say labels all the amino acid except C terminal amino acid, which is released as a free amino acid. So, only C terminal amino acid will be unaltered, rest all the amino acids will be labeled with the hydrazine. So, if we use chromatography, then we can identify, we can separate and identify this free amino acid, and which is the C terminal amino acid. So, by using this chemical method, we can identify only single C terminal amino acid. So, this is all about the identification of C terminal amino acid. If you have any query or confusion, please write it down in the comment section below. Thank you.